And as the last speaker, I am very excited to introduce nobody less than Matthew Collins, who will be talking about where we go from here. What is the future of paleoproteomics?
Yeah, thanks, Zandra, for that title as well. And thanks to the wonderful speakers beforehand. And thanks for organising this conference. It's amazing how many people we've got here. It's wonderful. Right, I am going to talk basically about these 6 ideas, about how we fit into linking disciplines together, the cultural challenges of working with other disciplines, the issues around complexity of the analysis we do, the problem of authentication, diagenesis sugars and Maillard, and the role of artificial intelligence. And by the time you see that last slide, we're almost over. Okay, the first one is thinking about the way that actually we tend to sit in amongst very many other disciplines. And it's really interesting to think about the kind of questions we ask. So if we're working in the area of paleontology, we want to know about the sedimentology, we want to know about the diagenesis, we need to work with zoologists that understand, and paleontologists, the context of the samples in which we work. All of those things are relevant in archaeology, but then you add on to that the taphonomy, why that particular sample got preserved in the place it got preserved in. Also, all the other contexts of the archaeology around it. What about the remains of material culture and that we can use to actually help interpret? And then when we come to history, it's kind of a very different world. It's kind of quite a sharp turn, I found, because with historians, we're looking then at direct evidence from text and how we can begin to integrate. And of course, that's what I've been doing a lot of recently in a project just now finished, an ERC project called Beast to Craft, which was using zooms, as Sam mentioned very beautifully earlier on, but to analyze parchment. And it's actually quite worrying and distressing to think that Sarah Fidderman, that really was the founder of the whole movement of barcode ecology, because she actually sits between 2 areas, between biochemistry and history, she's been unable to find a permanent job. And so I think this is kind of like a slight worry about the greatness of what we do in interdisciplinarity and the innovation. Sometimes we're kind of like ahead of the field. And I think the thing to think about here are, when we are working in any of those areas, how the different disciplines work, and particularly in this case, the humanities and the scientists, and the different ways they think about evidence and methodology, and the way we need to sort of think about the kind of languages you use and the context. And one of the things that you'll think about, actually, there's a lot of us, but actually we're a very rare breed. There's not many of us in the ways that we are fitting into these other disciplines. And I just mentioned those. We could include forensics on top of that as well. And actually there's very few of us and there's very many questions. And yes, we have the core methodology of proteomics, I guess, but we are answering so many different things. And so we need to find ways of communicating. And I kind of think of this in terms of the Arrhenius reaction, about how we crash together and how we react, the energy we need. And new opportunities arise at those points of collision. I think that's what's wonderful about things like PAASTA. People are encouraged to ask the simple question, the dumb question, because there are no dumb questions. We are a remarkably dumb community. We don't know that much. We're all learning really fast, and no one has a priority of knowledge. And then the other thing is, I think, and you saw this beautifully in Jesse's talk, we actually begin to link these unconnected bits of information and suddenly get these wonderful emerging pictures and stories. And so if we think about archaeological science, heritage science, we've won 2 Nobel Prizes for radiocarbon dating and for ancient DNA. And Sam did that lovely figure where she showed the increase in zooms. This is a plot I did for a talk I gave on Tuesday to a European Commission meeting. And basically what I was talking about was the speed of the rise in ancient DNA and heritage science. And yeah, that funny blip, you're right, that's Jurassic Park. So in other words, the way that actually it connects to culture. And for us, where are we? Well, that's paleoproteomics. So we started later, but we're growing quite quickly at the moment. Are we still in that takeoff phase? Or are we going to stabilize? And if you look, there is actually 13,000, so log scale, there are 13,000 papers that included the words ancient DNA together in Google Scholar last year alone. So really that has become a very dominant technology and has led to the Nobel Prize. We have to think about that though in terms of how we move forward and the mistakes that were probably made, the pressures, the secrecy, the burnout amongst PhDs, the way that museums are now increasingly close to the idea of just giving away their collections to one research group or another to do everything, and they want to be more engaged in that. And so I think that we have to worry about the way the control over samples and instrument has led to a toxic research environment. And that's really where all of these communities like PAASTA and SPAM and HAM have emerged to try to, you are the future, and you as early career researchers need to make sure that we don't allow that to happen in the case of ancient proteins. And it's not just, it's also whose culture, whose samples. We are working with the communities whose material this is. So that's just a rant, which I gave to the European Commission 2 days ago. In terms of complexity, then, we think about it, well, we heard beautifully from Sam about the power of barcoding. And really, I think it's fascinating to see how much, how she and others have really taken on Zooms and applied it to so many different research questions. And that's been really useful. And then Jessie mentioned this, and I think this is fascinating. She highlighted the way in which sex identification with enamel has led to so much innovation in the last few years. And like her, I was putting a list together. There's so many papers looking at new technologies for sex identification, because clearly there's real pressure in the community for doing it. And what we want to be able to do is do this reliably and cheaply and across as many tags as possible. And then the other question is in the area of phylogeny, which is a kind of well-established area, and one we have to think about quite carefully, because we've heard about the power of Zooms to discriminate. How much information do we actually get in terms of drawing out phylogenetic inference? We'll hear more about that in talks later today. And then there's sort of other interesting questions. A paper linked to enamel that came out a few days ago, identifying the possibility of getting immunoglobulins and therefore stress markers out of the enamel proteome, along with other molecules. The complex mixtures question that was touched on by Bea, how hard is to make that complex recipe and all the different components. And then finally, a thing that I obsess about a lot, the issues of diagenesis and the complexities, which I'll talk about. So those are the complexities. And of course, those complexities get ever greater with new technologies. We've already heard the name of Ockham, William Ockham, and I should put a link to the wonderful In Our Time recording of his philosophy, and then about this challenge of mixed protein sources. And in terms of novel technologies, they're about speed now, they're about resolution and about cost. And the things that we actually care about are in reversed orders. We want things which are low cost, accessible to us because we tend to have many samples. We want the highest possible resolution. And for us, because our samples are relatively smaller than others, speed is possibly less relevant to us, but speed does then link to cost. And we talked then about MALDI, we talked about Zooms, we talked about Bea, talked about ionization. But we live in this massive space of innovation and development. Look at that scale to 2022. And actually, we know Zooms is occupying a relatively small part of that overall area of innovation. So there's lots of things that we can be thinking about. And I think it's great to talk to physicists. Another really important thing when we come to shotgun proteomics is DDA or DIA. That is the question. And what we're talking about there is we are focusing mainly on DDA. And what that means is that we're actually looking at specific masses. And so we're acquiring data on specific intense masses. And that is very useful for the kind of de novo that Bea has just showed you. But what you don't do there is you don't analyze everything. And if you're working with much simpler systems, and spin is an example of DIA, we haven't mentioned spin yet, which is a kind of a new development over Zooms. The advantage here is you actually capture everything. At the moment, I'm worried about it because I am obsessed that we should be using de novo sequencing to do our identifications in shotgun metaproteomics, the challenges we have. And with DIA, that becomes very different. But it is now being used in ancient proteins, and it's something that we should be thinking about when we design our experiments. And then in terms of this challenge, what we're beginning to realize is that we have multiple proteoforms being produced from the same genes. we thought 20,000 genes in the human genome. We now identified 80,000 different ways that DNA has been converted into proteins. And the other issue, which has been mentioned by Jesse, are these complex mixtures, and Bear mentioned it too. We often are dealing not with a single organism, as illustrated beautifully by Sam, but with these complex mixtures. The question's already come up. How do we deal with those? And we've heard about William Bavockham. What William Bavockham is saying is, if you've got multiple peptides, then the simplest answer tends to be the right one. And this is the fundamental principle of many of the bits of software that we deal with. So here's the Max Quant statement about razor peptides. The peptides that match a protein group are assigned to the protein group with the most unique peptides. That's totally logical if you're dealing with a single tissue from a single organism. It becomes very challenging when you're working with complex mixtures. And you've seen this image before. This is an image where we are becoming obsessed with how much data is actually sitting there out on the archive and how much of it we're able to identify. And what you notice is the majority of these samples, the first two are large-scale modern samples, if you can see my arrow. And then the subsequent ones are ancient samples. And by and large, with ancient proteomes, we are doing poorly in terms of identification. And part of that is because the way we're doing our database searches, we tend to be using database-based searches, and we tend to be allowing for relatively little differences. And this is a study taking one simple protein, BLG, that Bharat and Ishmael have done together. And what you see is that you're seeing this is the number percent of the total proteome identified in the modern sample, 40 days degraded sample and 128 days degraded sample. And you're seeing is very, very little identification, depending upon whether you either use a small or a large database, triptych, semi-triptych, or non-specific searches. All of these are possible ways of analyzing this data. And what's remarkable, and something that really Ishmael's been pushing on, is using these open searches, where a much higher percentage of the peptides are matched. And really quite strikingly with pFind, it almost doesn't matter which setup you use, whether you use triptych or semi-triptych, you will get a much higher level of identification. And yet we've been tied many to Mascot and Max Quant because that's what we've been told to do, because that's what everybody does, and that's what we've all done. And so I think we need to be thinking about the way with ancient proteins, we need to be analyzing complex proteomics using shotgun proteomics in different ways. And one of the things that Jörn is now working on is an AI-first de novo sequencing approach, followed by an open search approach. So the AI de novo sequencing, that's what Bear was showing you, and then the open search is just what you saw in the second figure. And combining those together, we think we're going to get better results from ancient proteins. And then Jörn is working on a tool called Anubis, which is a deramified diamidate tool, which is all about trying to improve authentication. And this is going to be made available. It's going to be published very soon on Google Colab, so everyone will be able to use it. So again, this is all about accessibility. And then the question is, what are these things that we found deep in our ancient sample? Are they actually ancient proteins? And this is this problem of authentications. Is my sample real? And this is going to be a problem for us in the future. And we have various kinds of challenge. Replication, but that's really difficult because you need someone else with a lab to analyze your sample. And resequencing is one way we can do it. Checking our blanks, the contamination crossover that Bear mentioned. The appropriate molecular behavior. Does it seem reasonable that this molecule can be found in this context at this time? And that's down to diagenesis, to quantification of relative proteins. And think about it in terms of what is the context in which I found this? And what's hilarious about that, I think they're all sensible things. This is a very old idea. It comes from a paper published in the year 2000, a very well-known paper in Ancient DNA by Alan Cooper and Hendrik Poyner called Ancient DNA, Do It Right or Not at All. It's controversial in many ways, that paper. But the point is, actually, they are the things that they were thinking about 24 years ago, and we're thinking about now. And the authentication challenge is replication, behavior, and remains. Replication, I mean, this is this issue around, are we going to be collaborate, are we going to be collaborative, or are we going to kind of try and get all the samples and hold them ourselves and not share data out? And so that's one of the things I think, and what's great about the PAASTA community, it's all about collaboration, it's all about sharing. And it's easy for me to say that as a senior professor, but the problem is that damages, I think, our younger researchers if we don't collaborate. And so, yeah, so authentication, we've been trying to do it. It's controversial. People don't like it. And here's an example where I knew I was right. We spent a long time with Colin Smith, basically working out how rapidly collagen degrades in bones. Here was our survival. There's loads of radiocarbon dates. We know we're right. And then Carly comes along and says, yeah, but you're wrong. And so this is actually very exciting. It's exciting to be proved wrong. I was pretty convinced when I was working with Carly that we would be able to predict the limit to survival in Australia, and she just blew it out the window. Now that's really great, because what it means is there is some sites now in Australia where collagen preservation is blowing out these predictions. We need to know how and why, because we can look for other sites in the tropics and subtropics where we're going to get good proteomic preservation. So I think that's really exciting. Then the other thing, authentication in terms of associated remains. Does the data make sense? If you've got chicken eggshell, does it link to the fact we've got chicken bone? Here's an example from Ebola, where Jan will maybe talk about it more later. It's like how you actually make sense of the proteomic data. And the other thing linked to that, which I think is going to be increasingly valuable, is the idea of proteogenomics, where in this case, we have genomic data, for instance, in chewing gum, and we're now looking at the proteomics in the same chewing gums. Does the data all make sense? When we can use the genomics with the proteomics, that helps improve the database searches. The reason they're often so bad is because our databases are not fit for purpose. They're too narrow or they're too broad. Last kind of real worry is Maillard. And the fact is that we know there are sugars in our samples. We've only got one person, Kai, I think, who's now working on ancient glycoproteomics. It's a challenging field. But the reality is we know there are sugars present in almost all of our systems. And what they are doing is they're reacting primarily with lysine residues and arginine. Yeah, those two sites for trypsin digestion. Some really elegant work is now coming out using ultra high resolution mass spec. And this will be available, I'll mention at the very end, I think, in the new BioArc Riches platform that's going to be announced in October. But basically, using these techniques, it's extremely high resolution. And what you're able to do is identify masses to the atomic composition. You don't know the structure, but you know the composition. And here's some lovely experiments by Steve Larter's group, my old boss, looking at taking glycine, here shown in green, and starch, sugars, and essentially reacting these over time, and then following the composition of the oxygen-carbon ratios and the hydrogen-carbon ratios, and the nitrogen-oxygen ratios and nitrogen-carbon ratios. And what you can begin to see with this is that over time, you start to produce more compounds with larger number of nitrogen and oxygens. You're starting to form condensation products between the sugars and the nitrogen. We don't know what the chemical structures are, but we do know those things are forming. How do we think those through? And you come up with a concept of islands of stability. This is plotting oxygen carbon against hydrogen carbon. This is looking at organic matter in the world. This is geo-organic matter. And you see there are certain combinations of these constituents. And what Steve and his team have been doing is saying, at what point do we see these forming where we react different sugars with different plant starches in this case? We haven't got any really good papers yet on analyzing this in the connection with Maillard, with proteomes, but it's something that we should all be having in the back of our heads. There are sugars there, they are reactive, they are going to be affecting our results. And then last but not least, AI, and of course you probably gathered that AI generated quite a few of these damn images. But what I think is interesting about AI is AI loves shape. And so what you see here is a paper that's just been published as a consultant paper on looking at the shape of lithic artifacts. And this is proving fascinating. This is work of Aarhus University and others. And what they're showing is that when you look at the shape of these objects, the cultural boundaries given by local and national archaeologists break down. You actually see very, very different structures because, of course, they're tied much more to the morphology of construction. And AI can see beyond national boundaries and beyond national typologies. So this is very exciting. This is work on lithics. A lot of work is being done on lithics in this area. But I mentioned this thing called Richards. This is a large investment by the UK community. It's called Research and Infrastructure for Conservation and Heritage Science. I don't know if we can release my slides now officially until October when this announcement is made. It's pretty soon, and Jesse can speak to it more than I can. But one of the projects is looking at this idea of, yeah, we're getting all these archaeological objects, and many of us work on archaeological materials. Let's try and tie the 3D shape data to the molecular data. And this came from a presentation last week at the launch of this, which is the Biocultural Heritage Information in a Virtual Environment, Biocultural Hive. And this will be collecting over the next 5 years, lots of shape data, and they'd love to link this to Zoom's data and other molecular data. And in addition to that, there's another project which Jesse may want to talk about. This is why I don't think we can release this until October, but they have got large-scale funding to actually support Zooms and other kinds of biomolecular analysis at York. Now, this I think is only for UK, and Jesse can speak to it. But what is exciting to me is we've now got initiatives to look at molecular data and shape data and combine them. And so what is really important, we'll hear more about this later in the context of Zooms, is we need open and data, and we need inclusive innovation. So this was a talk I gave on Tuesday this week for IRIS about a thing called DigiLab. But the point about this is we have to think about our data as not being too undercooked or too overcooked, but just right, the Goldilocks of data. We need to have open access data formats, and the Zoom team you'll hear more from them about that. And the idea that we want to then link the AI, the ability to look at, for instance, at shape with data tools. And so, again, the thing I'm most excited about is looking at tooth shape data and enamel, the area that Jesse has mentioned as being extremely rapidly expanding at the moment. So yeah, in terms of future opportunities, lots of opportunities in the future built around collaboration, that we must make everything accessible and low cost, which is easy for me to say as a senior professor and harder for you to support. And then ethical and cultural considerations. And what we learn is that the more you share data, the better the data generally is. And now, as I say, interesting things around sex and animal bone identification through shape. Obviously, bringing in now the data on enamel will be fantastic. And so I leave you with this final point, that if we take the object as the starting point, and then we start to think about it, you start to be forced to bring in a whole variety of different thoughts and people, and to explain what that object is, what it did, and how it lived. And so, thank you all to PAASTA, to this opportunity to speak to you all, and to all the wonderful colleagues that did all that wonderful work, and nothing was basically mine, sadly. Okay.